Before we dive in, I want to give a quick shout out to the sponsor of this video, Subthesis. Subthesis is an all-in-one research platform that I personally use. It offers a full suite of free tools for literature review, data analysis, methodology selection, and manuscript preparation. Everything runs right in your browser with no login required, and all your data stays completely private on your device. If you enjoy this video, please consider liking and subscribing to the channel. It really helps me keep making content like this. You can also visit subthesis.com to explore their free research tools for yourself. Welcome everyone. Today I am going to walk you through how to use artificial intelligence to convert messy lab data into datasets that are compliant with the Findable, accessible, interoperable, and reusable principles. Whether you are a researcher, a data steward, or a leader overseeing research operations, this presentation will give you a practical roadmap for modernizing your data workflows. By the end, you will understand the key steps, the tools available, and how to get started in your own organization. Let us start with the elephant in the room. Lab data is messy. I have seen it firsthand, and I am sure many of you have too. We are dealing with inconsistent formats. One lab exports data as a spreadsheet, another as a document, another as a proprietary instrument file. Then there is the metadata problem. Most datasets are missing standard labels, units, or provenance information. On top of that, data lives in silos. It is scattered across shared drives, lab notebooks, email attachments, and personal hard drives. And here's the kicker. Researchers spend roughly 80% of their data handling time just wrangling and cleaning data. That is time not spent on actual discovery. So what are the principles that we are trying to meet? They were published in 2016 and have become the gold standard for scientific data management. There are 4 pillars. Findable means your data has rich metadata and persistent identifiers so that both humans and machines can discover it. Accessible means there are clear, open protocols for retrieving the data, including authentication when needed. Interoperable means your data uses standard vocabularies and formats so it can work across different systems and tools. And reusable means your data comes with clear licensing, provenance information, and meets community standards so others can confidently build on your work. Now let me show you the gap between where we are and where we need to be. About 80% of a researcher's time goes to data wrangling instead of actual analysis. Roughly 70% of published datasets lack sufficient metadata to be reused, and fewer than 15% of published datasets are truly compliant with these principles. Why does this matter? The reproducibility crisis gets worse without structured data. Funding agencies now mandate data management plans. Manual curation simply cannot keep pace with the exponential growth of research data, and human error at scale introduces inconsistencies that undermine trust in the data. This is where artificial intelligence comes in. Think of it as a bridge between your messy, raw data and clean, compliant datasets. The pipeline works like this. Messy lab data goes into an artificial intelligence-powered pipeline, and what comes out the other side is a structured, standardized, compliant dataset. There are 4 key capabilities that make this possible. Natural language processing and large language models can parse unstructured text, documents, and notes. Computer vision can extract data from images and instrument readouts. Machine learning classification can automatically categorize and tag datasets. And knowledge graphs can map relationships in your data to standard ontologies. Let us walk through the 4-step pipeline. Step 1 is ingest and parse. This is where artificial intelligence reads and understands all the diverse formats in your lab. We are talking about spreadsheets, documents, electronic lab notebook exports, instrument data in specialized scientific formats, and database dumps. How does artificial intelligence help here? Large language models can parse unstructured documents and even handwritten notes into structured fields. Optical character recognition combined with vision models can extract data from instrument readouts and images. The system can auto-detect file types and apply the right parsing strategy, and it handles all the messy edge cases like encoding issues, delimiter variations, and missing headers. Step 2 is extract and standardize. This is where the real transformation happens. Let me show you a quick before and after. On the left, you see raw lab data where temperature is recorded as 37 degrees Celsius in one place, 98.6 Fahrenheit in another, and just room temp in a third. The same inconsistency shows up with acidity measurements and concentrations. On the right, after artificial intelligence processing, everything is converted to standard units. Temperature becomes 310.15 Kelvin. Acidity is a clean numeric value. Concentration is unified to milligrams per milliliter. 3 key techniques make this possible. Named entity recognition identifies chemicals, genes, and measurements in your data. Unit normalization converts everything to international system standards. And schema mapping aligns your fields to community-accepted data schemas. Step 3 is enrich metadata. This is often the most overlooked step, but it is absolutely critical for making data truly findable and reusable. There are 3 areas of enrichment. First, ontology mapping. Artificial intelligence maps your terms to established ontologies, like those from the Open Biological and Biomedical Ontologies Foundry and schema.org vocabularies. This means your data speaks the same language as every other compliant dataset in the world. Second, persistent identifiers. Every dataset gets a digital object identifier. Every researcher gets an open researcher and contributor identifier. Reagents get research resource identifiers. Chemicals get international chemical identifiers. These identifiers make your data permanently findable and citable. Third, provenance tracking. The system automatically captures instrument metadata, processing parameters, version history, and operator information, so the full lineage of every data point is preserved. Step 4 is validate and score. This is where we close the loop and measure how well we have done. On the left, you can see a scoring dashboard. Each of the 4 principles gets a compliance score. In this example, Findable scores 92%, Accessible scores 88%, Interoperable scores 78%, and Reusable scores 85%. On the right side, I have listed the automated checks that the system runs. Schema validation against community standards, metadata completeness scoring, ontology term verification, persistent identifier resolution checks, license and provenance audits, and cross-reference integrity tests. These checks run automatically every time data flows through the pipeline, giving you a continuous quality assurance process. Now let me give you a tour of the tool landscape. I have organized the key tools into 6 categories. For natural language processing and large language models, you have options like OpenAI's models, Hugging Face transformers, Spacey, and LangChain. These handle text parsing, entity extraction, and metadata generation. For data wrangling, tools like Pandas, OpenRefine, Trifacta, and DBT help with cleaning, transforming, and deduplicating datasets. For domain-specific work, there are specialized tools. RDKit handles chemistry data. BioPython handles biology data. AIDA handles materials science. And the Investigation, Study, Assay Framework handles experimental metadata. For compliance assessment, FairShake, FUGI, the Evaluator tool, and the Metadata Template tool help you score and validate and certify compliance. For ontology and vocabulary services, the Ontology Lookup Service, BioPortal, Zuma, and the Open Biological and Biomedical Ontologies Foundry provide term mapping and semantic annotation. And for publishing your datasets, repositories like Zenodo, Figshare, Dataverse, and the Comprehensive Knowledge Archive Network provide persistent storage and discovery. So how do you actually get started? I recommend a three-phase approach. Phase 1 spans months 1 through 2. This is your assess and plan phase. Audit your current data landscape. Identify your highest value datasets. Select a pilot domain or project, and define your target compliance metrics. Phase 2 spans months 3 through 5. This is your pilot and iterate phase. Deploy the artificial intelligence pipeline on your pilot data. Train models on your domain-specific vocabulary. Validate outputs with domain experts and refine and expand coverage based on what you learn. Phase 3 starts at month 6 and continues forward. This is your scale and sustain phase. Roll out across departments. Integrate with your existing laboratory information management system and electronic lab notebooks. Establish governance policies and monitor compliance scores continuously. The key insight here is to start small, prove value quickly, and then scale with confidence. Let me leave you with 3 key takeaways. Number 1, compliance with the findable, accessible, interoperable, and reusable principles is no longer optional. Funders and journals are demanding it, and this trend is only accelerating. Number 2, artificial intelligence can automate more than 80% of the data curation pipeline, from ingestion all the way through to validation. This is not science fiction. The tools exist today. And number 3, start small with a pilot project. Iterate quickly based on feedback, and scale with proven results. You do not need to transform your entire organization overnight. I hope this has given you a clear picture of both the challenge and the opportunity. I am happy to take any questions you may have. This slide lists the references cited throughout this presentation. The foundational paper by Wilkinson and colleagues from 2016, published in Scientific Data, is where the principles were first formally defined. I have also included the Clark and colleagues' 2019 paper on the FairShake toolkit from Cell Systems. You will find links to the Go Fair Initiative website, the Open Biological and Biomedical Ontologies Foundry, the LangChain documentation, the RDKit Cheminformatics Library, Hugging Face Transformers, the Carroll and colleagues' 2021 paper on operationalizing the care and fair principles for indigenous data futures, and the Zenodo Open Research Repository. All of these are freely accessible, and I encourage you to explore them.